Hello again. The other topics is dried brown seaweed as a natural bioabsorbent of phosphorus of aqueous solution at laboratory condition. I am Mahmoud Hafezieh from Iranian Fisheries Sciences Research Institute with our colleagues Dr. Shahram Dadgar and Ms. Mahmoud Hafezieh. This is the world capture fisheries and aquaculture production from 1950 to 2080. You see the increasing of aquaculture activities in the world, and this is the prediction of global seafood consumption 2030. In the future, the farmed fish is raised compared to wild caught. Okay, what is our problem? When we have done aquaculture, we produce the waste. Of course, aquaculture is source of significant amount of the waste, which generally leads to deterioration of water quality, and of course removal of two important ions, I mean nitrogen and phosphorus, from aquaculture wastewater is an important environmental challenges. Okay, we have very nice picture from rainbow trout in our country, but for 1 kilogram of fish production, we release a different percentage of nitrogen and phosphorus, as you can see in this picture. If you fed the fish with 100% nitrogen input, 52% in fish body accumulation, 30% in case of dissolved excretion, and 80% as solid discharges. And if you fed 100 phosphorus input via feed to the fish, near to 33% in fish body accumulated, 21% in case of dissolved excretion, and 46% as solid discharges. In Iran, means in 2019, the total production in aquaculture in fish farm more than 520,000 tons, and actually the troutfish production more than 170,000 tons. Each ton of troutfish products released 150 to 350 kilograms solid dry weight. Nitrogen and phosphorus production per 1 ton troutfish is 7.5 and 45 kilogram respectively. Many aquaculture systems generate high amount of wastewater containing compounds such as suspended solid, total nitrogen, and total phosphorus. Today, aquaculture is imperative because the fish demand is increasing. However, the load of waste is indirectly proportional to the fish production. Therefore, it is necessary to develop more intensive fish culture with efficient system for wastewater treatment. A number of physical, chemical, and biological methods used in conventional wastewater treatment have been applied in aquaculture systems. These are the ways for removing nitrogen and phosphorus: ion changes, reverse osmotic, electrodialysis, biological methods, chemical methods, and surface absorption. But in this project we use a kind of biological method which is economical and environmental friendly. This is the system for removing the wastewater nutrient, and as biological method, we use seaweed as bioabsorbent, natural absorbent. As you know, the seaweed has so many importance as a human consumption, animal feed, fertilizer, cosmetic industry, green energy sources, and wastewater treatment. There are human consumption, animal feed, fertilizer used in cosmetic industry, biofuel, and finally wastewater treatment. This is the table of economic value of seaweed used for industrial consumption in different products, carrageenan, alginate, the hydrocolate from different kind of species, and this is the products in metric ton in USA per metric ton as $1. And what is our justification? We have near to 3,000 kilometer Iranian coastal line at the Persian Gulf and Oman Sea, and more than 150 different species, the seaweeds, and more than 10,000 metric ton seaweed biomass, which is 2,000 metric ton, especially sargassum set back from the sea annually. Here the map, satellite map of Iran, and here you see the coastal line of Sistan and Baluchistan, and this is the map of Persian Gulf and Oman Sea in Sistan and Baluchistan provinces. This is the Seaweed Taxonomy, and this is the different pictures of different species of seaweed from Oman Sea: ulva, Colpomania, Sargassum, Gracilaria, Hypnea, and other genus, Turbinaria, Gracilaria, Colperia, and others. And this is the setback seaweed from the Oman Sea to the coastal line. This is the huge amount of Sargassum erysiphorium, near to 2,000 metric ton based on Ajdari et al. 2007, the stock estimation, and other stock estimation near to 500 metric tons. As material and method, the preparation and characterization of biosorbent, we collected fresh sample of seaweed, Sargassum erysiphorium, from the coastal line of the Chabahar Bay, washing and dried and crushed to appropriate size, and after crushing, the seaweed's particle were passed through meshes with appropriate size to obtain particle, I mean 0.5, 2, and 5 millimeters, in order to determine the functional group responsible for bioabsorbent. The IR spectroscopy was performed. To this, 0.5 gram of seaweed was mixed with KBr for FTIR spectral analysis. Scanning electron microscope was used to investigate the morphology of bioabsorbent. The isotherm studies were performed in the solution with initial concentration ranging from 0.015 to 0.45 milligram per liter of phosphorus as optimum pH value for ion. After shaking the flask containing the mixture of biomass and solution for 60 minutes, the amount of phosphorus in filtered solution were analyzed. The bioabsorption equilibrium uptake capacity for each sample was calculated according to mass balance on the ion expressed in this equation. This is all other formula that we use for calculation of some factors, and as an experiment set up on absorption of nutrient by seaweeds. In the present study, the effects of several levels of pH 3.5, 6, 7.5, and 9, and finally 10 pH, initial phosphorus concentration 0.015, 0.15, 0.30, and 0.45 milligram per liter. Contact time 7, 15, 30, and 60 minutes. and particle size 0.5, 2 and 5 millimeter, and the seaweed particle concentration 10, 20 and 40 gram per liter were studied. You see the procedure for doing well, and the statistical analysis. We use Anova Analysis after a Shapiro-Wilk test for normalize the distribution data to determine the statistical weight of pH, initial P concentration, contact type, particle size, and seaweed particle concentration in P removal. Regression analysis was performed, and beta coefficient was used to determine the factor weight. Data are present as minimum plus minus ST. All analyses were performed in SPSS version 22. As a result, this is the FTIR spectroscopy of seaweed. You see the fluctuation of different groups. For example, the OH groups along with carboxyl group confirm the presence of carboxylic acid group in the absorbent. The presence of NH group along with carboxyl group might be attributed to the presence of amino acid group in absorbent. And this is the assignment of bands to functional group on the surface of the seaweed observed from FTIR spectroscopy. Okay, this is the SEM, the micrograph of SEM analysis of seaweed before and after absorption are presented in this figure. The micrograph clearly revealed the surface texture and morphology of the bioabsorbent with a large surface area. And this is the Langmuir isotherm of phosphorus equilibrium. Of course, this is the Freundlich isotherm of phosphorus. In this case, the R2 equal to 0.14, but in this case R2 equal to 0. more than 8. This is the parameters of isotherm models for adsorption of phosphorus using seaweed. Compare the Langmuir parameters and Freundlich parameters, and this is the graph for effect of initial pH solution in phosphorus absorption of the seaweed. The minimum in 3.5 and the maximum between 6 to 10 pH solution. This is the absorption as a percentage in different time, 7 minutes, 50, 30 and 60 minutes. The highest absorption percentage was obtained in 60 minutes. The initial phosphorus concentration significantly affected phosphorus removal from medium, as the lowest removal was related to the lowest initial phosphorus concentration. In this figure you see the absorption percentage based on initial phosphorus concentration: 0.015, 0.15, 0.3 and 0.45 milligram per liter. The highest was obtained between 0.52 0.45 milligram per liter. And seaweed amount significantly affected phosphorus removal from medium, as the lowest removal was related to 10 gram per liter seaweed concentration. However, there was no significant differences in P removal among 20-40 gram per liter seaweed concentrations. In this case, seaweed particle size significantly affected P removal as decrease in particle size from 5 to 0.5 millimeter resulted in increase in P absorption from 85 to 98%. And the regression analysis results are presented in this table. The model was capable to predict P removal using the predictors. The highest weight for P removal was related to seaweed particle size and seaweed concentration. The lowest weight was related to medium pH. As a conclusion, the present observation inferred that the Sargassum ilicifolium as a brown seaweed we have huge resources mainly in the Oman Sea coastal line showed a significant removal in nutrient, nitrogen and phosphorus. Hence, this species can be considered as a potential candidate for bioremediation of domestic and industrial effluent which can cause environmental pollution. It is concluded that the brown seaweed species not only can be cultivated in shallow coastal ponds used as bioremediant agent in the effluent treatment plants for conservation of nature and culture and natural resources, especially marine shrimp and fish aquaculture activities, but also after harvesting and drying also can be as a good bioabsorbent. Furthermore, the effluent cultivated seaweeds can be utilized as a feedstock for production of biofuel, biofertilizer, and additives for animal feed, which can reduce the cost of wastewater treatment procedure. This is the picture of Sargassum ilicifolium, and thank you for your attention.